It's
what is this software?
Vector NTI. Oh, this is Let me show you this. This is the symbol. Let me search. It's from Imagen. It's too expensive now. Vector NTI. So it's about $2,000 now.
To buy the program?
For one computer, for one software.
Wow.
$2,000. We bought this. We bought 7 or 8 licenses 10 years ago.
Yeah.
But they don't support it anymore. So whenever you change a computer, you reinstall computer, you lose your license. Now, our department, IT guy, they built 2 systems for me. So I can. This is new system. This is old system.
This is old?
Yeah, this is old system.
Now, but can you explain some of this one?
Explain what?
Explain, like I see locks P, and where do you put your gene?
Okay, let me show you. You know the CRISPR, right?
Right.
I have a CRISPR file here. They're all different of and there's a Cas9.
Yeah.
Okay, this is a Y-type Cas9 plasmid. This is Cas9 gene, and CMV. This is promoter. Okay, this is ampicillin resistant gene, and so neomycin. So basically, this is a Cas9. You can just electrocute this plasmid into the cells to express Cas9.
Now, do you put this? Do you put the DNA into those cells, or the RNA or protein? What do you put
DNA, just prep DNA plasmid. You purify the plasmid, then electrocute the plasmid into the cell, to the ES cells.
Oh, okay. And and that's pretty efficient, obviously, right?
Yeah, very efficient. Yeah.
And so, is there a selectable marker?
Yeah. Then you can select by neomycin, the selection marker.
Oh, I see. Yeah, there it is.
Here.
Yeah. Right.
Okay.
So you do that, and then that's CRISPR, Cas.
That's a Cas9. Then you need a guide RNA for CRISPR.
Right. How do you do that?
Yeah, that's okay. There's a CRISPR. Okay. This is called CRISPR uses guide RNA. You can buy this from Edgene, and then you can insert. Let me see. This is scaffold. So you can insert. This is U6 guide RNA. Where's the promoter? Oh, this is the promoter. Okay. Then you can see the sequence over here. Okay. This is the promoter. U6 promoter. Then you can insert your gene. There's a restriction site here. Then you can insert your gene, targeting gene.
What does it say? I can't read it. Is it BST?
BSA B1.
Oh, BSA B1. Okay, so is that the typical restriction enzyme that's used?
It's kind of weird. So it's kind of unique. So you can cut. But this we didn't use this one. We modified this. Our lab modified this CRISPR and make it a lot easier to insert the gene. But the idea is the same. This U6, then you have U3 site, then you insert your gene.
And the gene is how long typically?
Okay, let me show you the paper.
Because the gene probably is not like the full length gene at all, right?
Yeah. So there's a paper here. So this is
what paper is that? So
you must have read this paper before.
Oh, I see.
Yeah. So this is the principle for design the CRISPR. So you have you have a gene, and then you want to target sequence. The CRISPR, the Cas9, to recognize a PAM sequence, right? The PAM sequence and it's NGG. So you have so this is GNGG, and then before this, you just find the 19 base pair.
So you hunt for NGG.
You just find your gene which has NGG.
Right.
And then 1, 2, 3, 4.
Now, NGG, N is anything, right?
N is anything.
So just two.
ATC.
So really, it just needs two G's.
Yeah, just need 2 G. Okay, and then there's N, and then before this N, then you count 1, 2, 3, 4, 5, 6, 7, 8. I think it's 19. Then there's another G. So this one has to be a G also.
Oh, I see.
So that's a rule. That's it.
And
Then,
how do you then? Where's the gene at?
this is a gene, human EMX1 locus. Let me show you.
Is this so? This is the gene you insert. That little section.
This is the gene CRISPR you want to target.
Oh, right. But how much do you put into that vector? This vector.
Just this part. Yeah, that's a good question.
You mean just 25 base pairs?
Yeah, that's it. 20 base pair, I think.
Hmm.
Just this
So
part.
20 base pairs goes into there.
Yes,
Huh.
that's it. And then you ligate this base pair into this one, and then put the plasmid into the cells. Yes, cell, whatever.
This
cell you want.
plasmid.
Yeah, this
And
plasmid.
this will cut here.
You
That'll go to the gene. Hopefully, cut.
You synthesize this. You
Right.
just like primer. You synthesize this fragment.
Hmm.
Okay, and then insert into this plasmid.
And this will make RNA, right? Is that
This
right?
is plasmid. You put it into
Right.
the cell. The
Right.
cell will make RNA.
Right, the cell will make RNA.
Yes.
And the RNA will just be this little bit because of that SP6 promoter, right?
Yes, that's
Or
U6.
U6.
Yeah, what's U mean? Use.
I think
It's a
it's
ribosome.
yeah.
I think
So it makes
it's a
oh.
constitutive expressed promoter. So
And this is at minus 20. It says. It says minus 20. Does it already have the?
I think it's minus 20 probably before your insert gene. Probably I don't know.
Where and is the insert go into here?
I think so.
But and we got to know where. Is it not blunt end or anything, right? I don't know. Like is it or I don't know.
Oh, there's a protocol online. There's a very detailed protocol from Edgene. You buy this, they tell you how much to synthesize. I think, how much to synthesize, then how to insert this. I think I can find the protocol.
What are these things here? Like
This is
272? What's all these base pairs? That seems high. Why such big base
I
pairs?
couldn't remember suddenly. That's
Indel.
Indel. So basically, this paper, the CRISPR, wild-type CRISPR, there's the off-target issue. Efficiency is very high, frequency is very high. So and then this paper, they use mutant Cas9. The mutant Cas9, they can only bind but one cut. So a cut, but one cut two double strand DNA. One cut two strand, only nick cut one strand. So, but if you use two CRISPR, so use one CRISPR cut here, another nick here, one cut the whole thing, only nick here, another CRISPR nick here, okay. Then you make a double strand nick, okay. Then you can still increase your targeting efficiency. And
this just came out in 2013, right? This paper.
Yeah, 2013.
So it's kind of like this is the main paper that people use.
That's that's one paper. The original paper is one science paper from the church paper and the John's lab. This is from John's lab again.
Now, what were you going to do here? You were going to search for something else?
Right.
Edgene made a really good Edgene CRISPR. I can show you. They tell you how to use CRISPR. You see, you can cut, you can nick. If you want to buy a nick plasmid, nick kit, whatever, you just open this. There are many choices you can use.
Oh, right. And they sell it too, right? They sell.
It's just shipping fee. It's Edgene. They don't make money. It's very cheap. It's so one plasmid, I think, $120 probably, including shipping. So you can buy based on your purpose, whatever gene, what CRISPR you want to buy.
Is
There's
that
an
it's
instruction.
to target to a particular gene? Is that right?
Particular gene you have to design the unique the targeting sequence.
I see.
You have to put a unique sequence into the CRISPR.
So what's the purpose of all the different Cas9?
So this
Why do you need so many different kinds?
Yeah, the D10A is the mutant CRISPR is can only make a nick, won't cut double. Okay, there's some double mutant CRISPR. I don't know whether they have D10A. Oh, it should be on other kind. This is nick.
Oh,
cut
and
one
you're all
strand.
D10A.
All D10A. Okay, there's some activity. Okay, see this one? There will be some double mutant. So this color means hot. It's very hot. A lot of people buy this.
Wow, that's really nice.
This CRISPR last year they sent more than 6,000 CRISPR to the world. You think it's really hot? I don't know how long it can be hot, but right now it's still very hot.
It's gonna be hot.
Double mutant means where's the mutation?
There's a lot of hot
There's
ones.
two.
Cold ones.
This is relatively hot, but not as hot
Oh,
as this
I
one.
see. I see.
Yeah.
So why would which? How do you know which one?
Eurocat.
How do you know which one to pick? There's so many.
Yeah, based on your purpose, doctor. Based on what you want to do. If you wanna, let's say, if you want activate a gene,
Yeah.
so double mutant means it only band, won't cut anything. Only band a gene. Let's say you have a gene, you want to activate this gene. Okay, you design CRISPR to band this gene, and then put activation like a acetylation enzyme. Okay, put acetylation or put another VPCT4, which is from virus. Then you let this protein specifically target your gene, but it won't cut. Only bind the protein over there, then activate your gene. So that's great, you know. That's
It is something. It doesn't knock it out. It promotes
right.
it.
It doesn't knock out. It activate your gene.
Wow.
So
That's something.
yeah, that's why it's hot.
There was some paper. Yeah, right. There was some paper that had like simultaneous activation of up to 8 genes. Do you see this in Nature Genetics?
Gene?
8 or 6, I forget. Nature Genetics, 10. So maybe it was this using 10 different ones of these. But even
Right.
activation, there's so many. I guess it's pure mycin selection and neomycin.
Yeah,
but,
there's selection marker.
but why, you know,
Well,
if you want to activate one, is there one better than the other, like VP64 or is VP160?
yeah, I'm not familiar with this, but
Which one would you use?
I use VP64. We use VP64 right now.
I see.
So we want to, yeah.
Can you, and now what if you want to insert locks, 2 flock sites? Can you go back to the home page? Is there a way to do that? What other choices are there? This is,
Oh, the homepage. Okay, yeah. I don't know.
Yeah, yeah,
Interfere.
Not
Okay,
going-
that's inactivate gene. Okay, like if you wanna put a methylation, you put a you put a methylase.
oh, look, visualize. What's this?
Oh, that's visualized. I mean,
But
there's
look,
another
it
paper.
fused to GFP protein.
Yeah, use of GIP. It's hard for me to explain this.
Yeah, but it must insert GFP into a region of interest. This is really good.
Yeah, you can do that.
Fused to a fluorescent protein. Well, that's handy
Even-
because we got this new imaging scope.
Yeah, that's kind of people want to do. Yeah, and it's just a paper published recently by this. They use a CRISPR to visualize the chromatin
Remodeling.
remote. Yeah. That's the repressor gene expression. That's interfere.
Yeah.
Okay.
No.
That
Which is the hot one here.
I'm sure I don't know this one. I don't know what this means. we didn't use this. We are not working on repressing. We just activation.
So this must be 2, right? Or is it just non-conditional? Is there a way to conditionally interfere? That is, flocks,
Yeah.
the LOX-P site. Which ones put the LOX-P there?
Loss P, I don't know. Loss P, you have. I think you need to put use not necessary to use CRISPR. Just use the regular. If it's me, I can do really fast. Just use the regular insert loss P into the gene.